So it gives me great pleasure to introduce a colleague and friend of mine, Dr. Robin Green, who is the Saunderson Family Chair of Acquired Brain Injury. She founded the Tele-Rehabilitation Center for Acquired Brain Injury that integrates research and clinical care for people with acute brain injury, including concussion. She's a scientist in the Canadian Concussion Center, as well as at the Toronto Rehab. Her research focuses on longitudinal studies of cognitive, mental health, and MRI outcomes in brain injury and the development of interventions to improve outcomes. And so I will pass it on to Dr. Green.
Thanks, Carmela. So welcome back, everybody, to our afternoon session of the conference and the final part of our meeting. So for those of you who are just joining us, I have a couple of reminders before we get started. We ask you that all questions be put in the Q&A section, which can be found along the bottom menu in Zoom. And if you have any comments, please just put them in chat. If you're having any technical issues, please use the chat and our tech support team will message you to assist. And please feel free to put your cameras on if you like. We'd be happy to see you. I'm just going to jump straight into our first speakers of the afternoon. Our invited speakers are Dr. Oshin Vartanian and Dr. Sean Ryan. Oshin is going to be speaking first, and they'll be talking about psychological, physiological, and neurological effects of repeated low-level military blast exposure, and that will be data from the Canadian Armed Forces. And Oshin is a cognitive neuroscientist. He's a defense scientist at Defense Research and Development Canada, Toronto Research Centre. He's an adjunct associate professor in the Department of Psychology at U of T Scarborough and a cross-appointed graduate faculty member at the U of T. His research focuses on the effects of various operational and environmental stressors on the health, well-being, and performance of military personnel. And Sean is, Dr. Sean Ryan earned his doctorate in immunobiology from the University of Toronto. He completed an NSERC postdoctoral fellowship with the Defense and Civil Institute of Environmental Medicine and U.S. Army Research Institute of Environmental Medicine, examining biomolecular interactions between neuroendocrine immune systems under severe physiological stress and environmental extremes and their impact on health and performance. And today, he's a senior defense scientist in the Military Operational Health Group at the Toronto Research Centre and associate professor at U of T. And without further ado, Dr. Vartanian, would you like to get going then?
Yes, thank you very much for the very kind introductions and the invitation to be here. It's a real honor. As the title of our talk suggests, we're going to be talking about military blast exposure and the impact that it has on the health and well-being of members of our military. So Sean and I have been, to set the stage for this, so Sean and I have been sort of doing work on the effects of blast exposure on the health and performance of military members sort of for about the last 10 years or so. And the purpose of our talk is to essentially give you an overview of some of the findings that have recently seen the light of day across a number of different studies involving different kinds of groups and populations within the military, but to also discuss some very new findings that we're kind of currently digging up based on ongoing studies. So we'll present some really new data involving some of the questions that have sort of arisen in the course of our investigation into this very interesting and important topic over the last 10 years. Our talk is essentially going to be a two-part talk. I'm going to do the first part, and my focus is primarily going to be on the effects of blast exposure on neuropsychological functioning and some of the brain imaging results that we have. And then Sean's going to do the latter half of this talk, looking at the relationship between blast exposure and neurotrauma. He's going to be discussing some of the current work that we're doing, including multiomics. We have had quite a bit of work done on the neurological biomarkers of blast injury. And he'll also finish by talking about our current ongoing collaboration with KMH, looking at some of the possible links between blast exposure and chronic neurodegeneration. And most importantly, at the very end, I hope to have some time to hear from the audience about some of the ideas that you might have to kind of move our research forward. This is probably the most interesting part of the whole presentation for us personally. In the course of this talk, there are two groups that I'll be talking about quite often. These are populations that have been the primary focus of our studies over the years. So those consist of breachers and snipers. So to the general population, snipers will be more familiar. So there you've got blast exposure from a number of different sources, but primarily from recoil while you're firing a heavy gun. Breachers are a very interesting group because they specialize in this tactical maneuver to gain entry into enclosed spaces during operations. So what a breacher will do, you can sort of see an image there. There'll be an enclosed space that the military members need to gain entry into. So they get a lot of training in planting specific kinds of devices and blast explosives of different strength, being able to cause a detonation and then to actually enter that space. Now, in the course of training for breaching, they tend to do this in stacks. So I hope that that picture stuff gives you a sense of how this might work. So they have to take into consideration all kinds of environmental and geometric factors. The explosive is detonated, then they have to actually enter the space. Now, in the course of training, you'll be exposed to many, many, many blast events of this kind. And what sort of typifies what we'll be talking about today is primarily an focus not so much on very large blasts that occur in theater, but exposure to repetitive low-level blasts in the course of training. And I'm very happy that Ben Dunkley went ahead of us because he already touched on many of the major themes that we'll be talking about, having to do primarily with comorbidity between for example, mild traumatic brain injury and post-traumatic stress disorder as a function of blast exposure. So this would be a typical study design for what we tend to do. So these breachers tend to train typically for about 4 days, and the trainings will be kind of happening in the course of the year. And in the course of those 4 days, they'll be exposed to a very heavy dose of repeated blast, trying to breach different kinds of spaces. They could be indoor and they could be outdoor. And so what we typically will do is we'll acquire a lot of baseline measures from them. These will be the standard neuropsychological assessments, MRI, a lot of measures on background health. And in the course of the 4-day training, they get tested on neurocognitive measures. Of course, we look at balance and ataxia because the vestibular system is very heavily impacted by blast exposure. So you can typically see acute effects on balance. And we look at things like postural tremor. Now, what you also see in this image at the bottom is a blast gauge. So these were developed in the US about 15 years ago. We use them pretty much for all of our studies. So typically a breacher will be equipped with 3 blast gauges, and these give you continuous measurements of blast overpressure in the course of training. So this gives you some kind of a quantified measure for the amount of overpressure the person is exposed to, which you can then correlate with different kinds of outcome measures of interest. And then when the training is completed, we again acquire MRI and a number of other measures. Now, in some of the other studies that I'll be talking about today, we have, for example, taken a cross-sectional approach. So in these studies, we will go to different kinds of Canadian Forces bases. We will recruit breachers and snipers. And then for every single person in our sample, we also have a sex and age match CAF control. So CAF stands for Canadian Armed Forces member. As you can see, we administer a very large host of measures, again, background health measures, neuropsych assessments, neurocognitive testing, balance and ataxia, hearing, blood and saliva, which of course Sean will be talking about, and visual-motor integration. We've been using this very interesting task that was developed by Lawrence Sergio at York University, which has proven very sensitive to the effects of concussion in athletes and sports, which requires a person not to do cognition or motor movement individually, but to actually combine them towards a response. And we find that this is really sensitive to the effects of blast, as we've shown before. So across many of these studies that we sort of did in the first 10 years of work in this area, these were some of the findings that were reliable. And what I mean by that is we found very similar effects across a number of different groups that do breaching under different kinds of contexts. So we very reliably see effects on post-concussive symptoms. So the instrument that we use most often is the river mead. We see greater difficulties in musculoskeletal function. Interestingly, when this work first started about 10 years ago, we hadn't really opted to look at musculoskeletal function, but the operators themselves noted that this is something that we're suffering from and that this was really worth looking into. We also see lower levels of self-reported health. So for example, administering the RAND SF36, they tend to report lower levels of physical and psychological well-being. We see higher rates of tinnitus, that's ringing in the ear. Also, not surprisingly, when you're exposed to blast, your ears are affected. We see difficulty in visual motor integration, which I've noted before. And then with MRI, we also see correlations between blast exposure and a gray matter in the prefrontal cortex. So we've done this in a number of different ways. So we collect structural MRI data from breachers and sex and age-matched calf controls, and we can do basic comparisons. But we also have looked to see whether using different kinds of multiple regression models corrected for different kinds of background factors, there is a negative correlation between gray matter and the degree of blast exposure, which we've also shown. And we also see alterations in neurological biomarker profiles, which Sean will talk about in a few minutes from now. So this is sort of the background that really motivated the work that we're doing right now. So we're running a major study for the Canadian Armed Forces, which essentially has two different goals. One of them is to explore more fully the impacts of blast exposure on health and performance, and also to see whether there's a need to establish a longitudinal health monitoring program so we can track our members who are exposed to blast in the course of their careers to see when the risk of developing different kinds of health issues is raised. So again, motivated by our previous studies, we have a large host of measures that we typically collect. So a typical data collection will consist of about 4 to 5 hours of data collection per person. Background health, neuropsych testing, neurocognitive testing, hearing, extensive brain imaging, and of course, biomarkers. So as I mentioned before, I'll be focusing on neuropsych and fMRI data, and Sean will do the blood and saliva component later. So one thing that really motivated our current work is something that Ben actually talked about earlier in his talk, which is this comorbidity that you see between post-traumatic stress disorder and mild traumatic brain injury. So this has been reported many, many times across studies, especially from studies coming from the US. And that's perhaps not surprising, because typically when a person in uniform is exposed to an event in theater, a major blast event that could be causally linked to mild traumatic brain injury, the event itself is also emotionally traumatic. So there's a good reason to believe that when you collect blast exposure data from members in theater, that you're going to see comorbidity between post-traumatic stress disorder and mild traumatic brain injury. On the other hand, the context that we're focusing on right now, which is breacher training, is somewhat of a unique enterprise, because here it might be possible to dissociate those outcomes in the sense that you'd expect to see post-concussive symptoms, but perhaps not mental health issues, because the blast exposure is not necessarily linked with a traumatic event that might have emotional sequelae. So in one of our early studies that we published a couple of years ago, so this is where we administer the Riverme to breachers and a group of sex and age calf controls. And of course, as you know, the Riverme can be scored in a number of different ways. So this is two methods that we use for scoring, looking at the immediate effects of post-concussive symptoms. That's RPQ3. So these are things like headaches and nausea and vomiting. RPQ13 is more the long-term effects, things like, for example, difficulty in concentration, sleep disturbances, and so forth. And as you can see, breachers report higher levels of those more long-term effects. Another way to parse the same data would be to look to see whether the effects are cognitive, emotional, or somatic in nature. Again, we see that there are statistically significant differences between breachers and controls when you look at the cognitive and emotional components of post-concussive symptoms. So in this early series of studies that involved data collection across 4 years, we didn't necessarily have a mental health issue measure, but we did have the SF36, which has the emotional well-being subscale, but we didn't see any kind of differences between the two conditions on that. So here we thought that there might be reason to probe this a bit further to see whether there was actually a way to distinguish between post-concussive symptomatology versus emotional issues in this particular population. So in a follow-up study, we went to a different Canadian Forces base. So this is the acronym that you see up there, and I've limited myself to only 2 acronyms, even though in the military, this is a very popular thing to do. So the CFSME stands for the Canadian Forces School of Military Engineering. So these are our military engineers who have a particular group that specializes in gaining entry into enclosed spaces and does loads and loads of breacher training outdoors. So here we administered, as before, our Rivermead, and as you can see, we saw a very similar pattern. Again, breachers are reporting significantly higher levels of post-concussive symptomatology. We also administered the PCL5, and we didn't see differences between the two conditions, even though you can see there's a trend in that direction, but the difference was not statistically significant. So we thought that's kind of interesting. It sort of mimics overall what we had seen before. Perhaps there's reason to look into this somewhat further. Now, in the course of that particular paper, one very bright person whom we have as part of our team is Alex Di Battista. I'm sure he probably works with many people who are on this call right now. He did some very interesting multivariate analysis of that particular data set for us to kind of really drive home the point that you can't really look at breaching individually, but you sort of have to look at the overall context within which breaching is measured. So what you see at the top panel are items that distinguish between breachers and non-breachers. And the two blue bars that you see there are elevated scores on the Rivermead in terms of the immediate and more long-term effects. In other words, if you're a breacher versus a CAF control, you're going to be having higher post-concussive symptoms. But you can cut the same data by looking to see whether the same people are reporting a concussion or not. And then, of course, you again see elevations in the Rivermead, but then you also see elevated scores on musculoskeletal difficulties, okay? And another way to parse the same data would be to see whether members in the same sample have been deployed to a war zone in the past or not. And something very interesting emerges here. What you can see is that the Rivermead scores are still relevant, but so is the PCL5. In other words, there's reason to believe that when the distinguishing factor is war zone deployment, it's not only post-concussive symptomatology, but also possible PCL5 scores as a function of what might have been encountered in theater. So in terms of our ongoing study right now, so we are about 90 participants in. We hope to run over just 100 participants. So these are, again, scores that you see on the Rivermead between breachers and snipers versus controls. And we see significant differences on every single subscale except the somatic one. So it's very similar to what we've seen before. And kind of like previous studies, we also administered the PCL5, but we broadened our measures of mental health indices by also including the brief symptoms inventory that sort of gives you measures on depression, anxiety, and somatization. So again, we don't see differences between the two conditions on these mental health outcomes, but we do see differences in terms of post-concussive symptomatology. So it looks like overall that it might be possible to distinguish between mental health issues and post-concussive symptomatology when the focus is on breaching environment where the events that lead to blast exposure are not necessarily traumatic in nature. Okay, so a quick 2-minute run through our MRI studies. So as I mentioned before, we do structural MRI, resting state fMRI, and task-based fMRI. We do most of our scans at the Royal in Ottawa, where most of our members are located, but we also do brain imaging at York University. So one of our tasks that we administer in the fMRI is a working memory task to see whether long-term exposure to blasts in the course of operations and training might have an impact on working memory function and the associated neural correlates. So everyone's very familiar with the working memory system in the brain is primarily frontal parietal. So we look to see whether there are going to be differences in the neural systems that are activated within the frontal parietal system as a function of a person's breaching history. So here we've got 3 groups that you can see on the left-hand side. So new recruits are people that have just come into the unit that are hoping to eventually become breachers and snipers. Okay, so this is sort of one of the earliest time points at which you can capture a proper naive control case for the particular person going forward. CAF controls are members in the military who are not breachers or snipers, and then our breachers and snipers are primarily experienced breachers and snipers who've been engaged with blast work for a long time. And what you see are the neural activations associated with 1-back, 2-back, and 3-back. And you can think of 1-back, 2-back, and 3-back as a gradation in difficulty of working memory demand. And what we see here is that you see significantly greater activation in the frontal parietal system when you are looking at breachers and snipers compared to CAF controls and new recruits. So typically how these data are interpreted is in terms of some kind of a reduction in neural efficiency. In other words, when a task becomes really difficult, that for people who have had exposure to something that you think might impact their functioning, a greater recruitment is necessary for carrying out the same task when you correct for level of performance. I just want to mention quickly that neural efficiency is a difficult measure, is a difficult construct to isolate and measure directly. So this is one of many possible things that could be going on, but it's consistent with that overall interpretation. And at this point, I'm going to switch over to my much more capable colleague, Sean, who'll be talking about the brain biomarker results. So I suppose that I'll be stopping my share and I'll go on mute. Sean, I think the floor is yours.
Thank you so much, Oshin. Always a pleasure to follow you. You set the stage so nicely. I will just ask everyone if they can hear me and see my slides.
We can.
Great. And thank you very much to the organizers, Dr. Tatter and Dr. Tartaglia and Dr. Green for the nice introduction. It's a pleasure to be here. And fortunately, a lot of the speakers have covered a lot of the material. So I do apologize if I gloss over or seem to some of the slides that I have, because everyone has done such a good job of providing a lot of the background. This slide I often put up as a bit of a, bottom line up front, because, you know, when, as Oshin has articulated very well, when we're dealing with military exposed to blast overpressure, it's a somewhat different entity. And even though we've been doing research for, wow, 10 years already, time goes quickly, we're still really scratching the surface, I think, in terms of trying to understand this in comparison to entities like mild traumatic brain injury or even concussion. Because I'm reminded quite often, particularly by our American colleagues, that what we're dealing with here is really a subconcussive impact, although it does certainly have measurable performance and health symptomatologies and decrements that we've heard about. So I put this slide up really just to say that there's a lot that we don't know, of course, but there are some emerging findings, some of ours and certainly some of the international work. This whole area of research has sort of been catapulted in the spotlight in the last 4 years, I guess, when the US Congress mandated for our counterparts in the US that they test either annually or biannually all military members who have exposure to heavy weapons and blast. And as Oshin mentioned, really it's the operational community that has come to us, self-identifying with a lot of the problems. So I put down here that blast intensity and frequency matter, and that we're really dealing with the lower limits of blast exposure here, as opposed to roadside bombs and those kinds of things. So we're looking for sort of a dose response effect if we can find it. Just because we're standing on the shoulders of some great work that came before us, although it's only 2013, this is really kind of a seminal piece of work that many know about, led by Dr. Kamamori's group out of Walter Reed at the time, and Dr. Charmaine Tate from New Zealand. And it looked at the population of New Zealand breachers during heavy training. They brought in Dr. Kevin Wang, who was then with the Banyan biomarker group. So it was really the first attempt to correlate some of the neurobehavioral, neurocognitive outcomes with the biology. There was no neuroimaging at this case, and a lot of the initial findings, they were quite weak. They didn't show a lot of response, but there were some significance with particular biomarkers, and that stimulated, I think, more interest in the field. I mentioned Greg Elder and James Stone and Stephen Allers, because again, they're a real powerhouse funded by the US Navy and have done a lot to put low-level blast exposure on the map in terms of a problem for the military. And as we'll hear about some of the longer-term sequelae that may be associated with neurodegeneration. Some of the usual suspects, most of us are familiar with things like S100B, glial-fibrillary acidic protein, ubiquitin carboxyl terminal hydroxylase. I think I only rival the military as an immunologist in trying to include a lot of abbreviations. So sorry about that. There will be some coming. But these markers are putatively associated with blast exposure, and there have been a series of studies building over the last decade. But I wanted to just emphasize that the samples are small in size. A lot of the conclusions are preliminary. And a lot of the findings require kind of standardization across trials. And that's what we're aiming for now through some NATO panel work, where we're hoping to work more closely with the international community to set up multi-site studies, standardize with basically standard operating protocols and common data elements, as we all strive to do in this space. I would mention, again, there's very little evidence on the human side. There is animal studies to show in blast models that there's evidence of CTE, but I would say that the evidence base to date for long-term neurodegeneration, in particular in military, is weak, although we suspect it is a problem based upon symptomatologies and anecdotal evidence at this time. Again, there's been some follow-ups to the initial New Zealand breacher study, but all of these studies kind of led to a RAND meeting in 2018, where I think the overarching conclusion was at that time that there was a distinct paucity of data, human data, to say whether or not blast neurotrauma was actually a problem. So that has allowed us to continue with some work. Oshin talked a little bit about the work that was published recently, where we were looking at mainly the neuropsychological functions. I'll hopefully get to some of the biological data that's associated with the CFSME breacher cohort that Oshin mentioned, as well as some of our other CAF populations that we're studying. Just put this up for context, because I know most people are familiar with the left-hand side with moderate, severe TBI, mild TBI, and the associated Glasgow coma scales or outcome scales. What's striking, though, is that on the right-hand side, where we're dealing with subconcussive neurotrauma, we're not really even dealing with something like a competitive athletics, where you have a collision sport. We're dealing with PSIs that are quite low. So those blast gauges that Oshin was showing, typically they're supposed to record below 4, but there is evidence that in repeated training, values can go up above 10. So it's really these repetitive low-level blasts over somebody's career, over their lifetime, mostly during training, because the mantra really has been, and still is to a certain extent with certain groups, we train like we fight. So they don't like to dampen down the intensity when they're working with explosives or heavy weapons. So these are the issues that we're dealing with in this space. And some of the biomarkers are indicated here, and I'll talk more about them. But the other critical thing is that a lot of the people we see, they're not diagnosed with anything. They're symptomatic, and they have some concerns. So what do we know really about the pathophysiology? This is a recent review from Frontiers in Neurology that kind of conceptualized everything nicely. Again, it depends on whether we're talking human or animal work, but we know from various studies that there is evidence of axonal and other brain cell injury. There's some mitochondrial dysfunction. Obviously, it changes in synaptic reuptake and neurotransmitter function. And certainly, and perhaps even very importantly, neurovascular impairments. We've done some work that's not published yet to look at matrix metalloproteinases and other endothelial factors, which seem to be quite impacted by low-level blast. An area that's just emerging more is to look at the longer-term effects. And so, some of the symptomatology, sorry about that, that we're seeing are really the common things that you would see: subconcussive syndrome, headaches, the fatigue, the comorbidities were neuropsychological outcomes, PTSD, depression, and a lot of what Oshin mentioned too in terms of the auditory and visual issues. The visual motor test that was done by a colleague at York University was one of the strongest associations, which is, when you're having to think and move at the time. It seems to be a real problem for some of these individuals with a lot of blast exposure. So leaping into kind of my world, if you like, where we're trying to tackle this issue on multiple fronts with a kind of a multi-dimensional approach. Multiomics is kind of a buzzword these days, but it simply means we're looking at proteomics in different ways, both small and larger scale. So we use technologies that are, immunoassay based, like Ella, which is a small microfluidic device where you can run stuff on a cartridge that's very quick. It's more amenable to point of care. We're also using SAMOA for some of our neuroproteomics, which I'm guessing that a large number of people on the call are familiar with or maybe using SAMOA. And more recently, I think we heard it mentioned earlier in one of the talks, there's a technology out of Sweden called the proximity extension assay, or it's made by a company called Olink, where you can start to approach several thousand proteins harnessing the power of next generation sequencing. We are also using this for our genomics. We want to know whether there's alterations in gene expression that are perhaps predictive of somebody's risk profile, or if they would correlate with some of the proteomic changes that we're seeing. Also, some of the epigenetics, we have a large number of samples that are just now under analyses, and we don't have the results yet, unfortunately. We're also looking at single nucleotide polymorphisms, again, to help predict those who might be at risk, at greatest risk, and looking more prosaically at saliva for microRNA profiles that seem to be indicative, at least in concussion. And there's some work to show that they may be instructive or informative with blast neurotrauma. Also, we do a lot of work in-house with flow cytometry, so we're hoping to expand the repertoire to look at cellular immune function with low-level blast. Again, a busy slide. I just put this up really to highlight that we have a lot going on. Most of what we are doing with the Canadian Armed Forces is really geared towards, first of all, detecting whether there's a problem. Can we monitor it? Hopefully we can mitigate it before it becomes a serious clinical outcome. And we're also looking at treatments, both from the standpoint of nutraceuticals. Oshin and I are involved in a study right now at the University of Toronto that's going to look at concussed athletes using omega-3s and other micro and macronutrient formulations, both prophylactically, but also as a way to track their recovery trajectory to see if there's any help there. I'll talk more about the tau imaging work that we're doing in collaboration with Dr. Isabelle Poilot and some really bright graduate students. Carmela's involved. It's really the first study to look at possibility of tau deposition being abnormal in military members with career blast exposure. I'll move on. Again, this slide is instructive, I think, from the standpoint that it points out that there's quite a lot of protein biomarkers out there. None of them are perfect. And what we're hoping is that there is a panel of markers that will tell us something in a kind of pathoanatomic way about which cells may be most affected, which ones are altered by looking in biological fluids. Of course, with healthy individuals, it's very hard to get CSF or direct brain readouts, so we are constrained to the peripheral blood. But as you'll see, there are some emerging patterns with some of the usual suspects, including GFAP or glial fibrillary protein, some of the neurofilaments more on the light side, axonal injury related, and also some of the long-term neurodegenerative markers like tau and phosphorylated tau. And we're using, again, as I mentioned, the SAMOA technology, which claim to fame is it can get down to less than 1 femtogram, so high sensitivity and good reproducibility, or as good as it perhaps can get. I put this one up, again, this is more just an overview of what we're doing. I'll get to some data, but this is a really encouraging area where we're just starting to analyze the results. When you look at the immune response, so cytokine expression, immune cell activation in plasma, what you're seeing is really kind of an echo of something that happened previously. So what we're doing here is we have this standardized way of stimulating immune cells that doesn't require centrifugation. You just culture it overnight in a heat block, and you get very robust release of a lot of the immune markers that sometimes are quite low in ostensibly healthy people or those who have a mild brain injury. So you not only get more data, but you get an actual readout of some of the functional activity of the immune system. So we've been collecting this for a few years now on our blast exposed population and are just starting to analyze that. This is some of the first data that was kind of striking when we were again working with the breachers from Gagetown, where we looked at a broad panel of about 15 markers. Again, most of the acute, subacute markers, and so on. And we found roughly a little more than half of them in a cross-sectional comparison with breachers with high exposure to blast and trainers, those who are on the range all the time, several days a week for many years over a career. they all had constitutively high levels of a lot of these neurological injury markers in the blood. So this data was quite compelling, even when we were looking at markers like total tau and TDP-43, which again are related to longer term neurodegenerative outcomes. We were quite struck by these results. So it spurred us on to do some further research. Again, Oshin mentioned one of our great postdocs and now defense scientist, Alex DiBattista, who did some partial least squares regression for us here to look at when you control for things like age and recent exercise, which is a physiological stress or perturbation, a lot of these markers were still robustly elevated in breachers versus controls. This is more recent data. We haven't quite finished all of our work with the breaching community we're working with, but this again was quite striking. Some of the more acute markers like neuron-specific enolase, GFAP, were elevated relative to healthy military match controls, even prior to heavy weapons exposure. So this group undergoes a lot of sniping over several days. They also have small arms fire. And it's not just the recoil that will get them in terms of the biomechanics of that. If you watch it in slow motion, it's quite powerful to see the recoil of these heavy weapons, but it's also the muzzle blast, these small, what would be relatively small blasts over a long period of time that we think are impacting them neurologically. And when we visit with some of these individuals, it's quite striking. Some of them are very young in the earlier part of their career, and they're already having to hang up their sniper rifle, so to speak, because of ongoing either musculoskeletal or other neurological concerns. So it is a big problem because our military, as we know, is quite small. And you invest a lot of money in these individuals. So there's a lot of interest in tracking this. And we're hoping to do some more acute studies with basically using dried blood spots and other point of care testing so we can get some readouts right on the range. These ones were after they returned from the training session. Now this slide I put up, and again, sorry, there's a lot of abbreviations, but essentially towards the left you have controls, and then you have some individuals who haven't been in the military very long, and then you have breachers, snipers, pilots, and on the far right-hand side, you have those who've actually been diagnosed with a sport concussion. So I wanted to put this up for perspective because I think we all know that when we're dealing with biomarkers, different ways of measuring, different platforms, there aren't very good control ranges. So some of these are on a log scale, and you can see there's several orders of magnitude difference between some of the concussed athletes and some of the other individuals. So even though we're seeing subtle changes, perhaps in a breacher versus a sniper versus a healthy person, we have to look at it in the context of what this outcome may mean clinically. And I don't think we're there yet in really understanding what might be fairly minor perturbations versus more extreme changes. So that's really the purpose of this slide. This one just shows, this is from a colleague on a NATO panel, who put together a nice retrospective kind of overview of the number of studies that have looked at neurological injury markers. And you have a little more than half that have shown significance. However, the challenges are still that there are many different measures, many different, it's all about the timing, the kinetics of when you take it, is it in a controlled environment where the subject's resting. So a whole host of pre-analytical factors that I think we still need to sort of harmonize to make some real progress in this domain. And this one, I won't spend too much time on it, but we have had access over the years and did do a follow-up study where we looked at all the way up to subarachnoid hemorrhage on the right-hand side, hemorrhagic shock, severe TBI. So again, this just accentuates versus healthy military who have blast exposure on the left-hand side. Even though there are differences relative to controls, the RC, the second column is actually exercise. So you can see that physical exercise stimulates the release of these so-called putative neurological markers, which is challenging. And then you have breachers here. So, but compared to very severe insults, in many cases, the marker levels tend to be quite modest. So again, this is something that we're interested in following up on. I'm going to switch gears a little bit if I have time. I know I have a lot of slides. So if you need to pull the hook out at any point, you can. But I just wanted to touch upon our work with the long-term neurodegeneration, and it's been very well articulated by Dr. Garcia and others earlier in the program that, you know, we recognize the post-concussive syndrome, chronic traumatic encephalopathy in sports, but we don't yet know really what the correlates are in the military. Again, and it's partly because we're dealing with this subconcussive phenomenon. There's certainly evidence in sports. We all know from professional football and boxing that these individuals are at much, much greater risk because of repeated head trauma. And the animal studies point to that in military members and humans. But I think there's still some work to be done before we're sure about this. This is really, again, just to show some of the structure of tau. As many of you know, tau is a normal protein that supports the microtubule in various tissues and neurons and so on. But with repeated head trauma, it can become altered, and it can be actually released and propagated to some of the deeper brain tissues, where it starts to, of course, cause issues of cell death and neurodegeneration, and then a lack of communication between neural networks and so on. And this entity is very similar, CTE, to what we would see in Alzheimer's and other types of dementia, although it is distinct. So just to say a few words about what I think is going to be an important study that some of us are involved in with Dr. Boileau at CAMH. We're taking the most senior members of the Canadian military who have 20 years or more exposure to low-level blast and weapon systems. And we working with Isabel and her colleagues, just blocking on Neil's last name, but a terrific group at CAMH to use the tracer that many are familiar with, the flataucipir, which binds putatively to tau deposits within the brain. So it is a bit of an in vivo way to look at the potential for tauopathy and the potential risk of CTE. We can certainly argue that later on. But I think it is important from the context, there's never been a study in the Canadian military to look at this, and there are very few well-controlled studies across the globe. The US has done some work, but they tended to look at more at those who had been exposed to IEDs, large blasts, not so much the repetitive low-level exposure. So we're hoping to get at least 30 experienced members who have exposure, a healthy age-matched control from the military, and we're also hoping to access some historical reference data for civilians. So this, if successful, this would be the first study perhaps to also compare central and peripheral measures. So we're looking at things like total tau, p-tau, A-beta 40, and some of the other long-term neurodegenerative markers in the blood. This study is really being headed up by a grad student, Shamantha Laura, who's working through IMS with a few of us, and she's done a terrific job with a very difficult population to recruit, not to work with. The guys love coming to the study, but with their schedules and deployments, it's extremely challenging to have them come to CAMH. But we're about halfway through. Some of this data has been, without any controls, has been looked at in terms of the correlates between tau deposition in the frontal cortex, temporal regions, whole brain cortices, in comparison to years of breaching exposure. And right now, with the early trends in about 25 individuals, we're already starting to see some of the data indicate that there is a correlation with exposure. It'll be very important, I think, and interesting to look at the levels versus controls who either have had no blast or very little blast in the military. So I won't go through all these conclusions, but essentially, I think where we're at now is that we need to standardize our studies across centers. We need more data, even though we've been doing this for quite a while now. We're hoping to wrap up, as I mentioned, and Oshin mentioned, some very large studies where we have a lot of blood, saliva. We're looking at everything from genomics all the way through to proteomics, and I think that'll be instructive. The US and other groups are doing the same. And then I would hope that we're going to move to perhaps to some standardized protocols, invoking some AI and machine learning algorithms with a little bit larger data set I really do, as others have remarked today, think that is the future in this area, and that we need to do that to make real progress. So I will leave it there for questions. Thank you.
Thank you very much, Sean and Oshin. Those are amazing talks that you gave. We're actually running over time, but what I'm hoping is that we can come back during the panel at the end to some of the great themes that you brought up, and maybe you can field some questions then. And if people have questions for Sean and Oshin, please put them into the Q&A or into the chat.